Okay, good afternoon, colleagues. Today I would like to present our work, Prediction of Toxicity Endpoints, as a pathway towards minimizing risks in drug development. So drug discovery scientists face a fundamental challenge: how to find a drug molecule that is safe for humans. And here comes the first problem. More than 300 million structures have been documented. However, other than how they were obtained, we know little about these structures. Moreover, the number of chemical structures that can potentially be synthesized is estimated at 10 to the power of 60. How to find such single molecule among the vast chemical space that can help in treatment of the most serious diseases? So probably we need to somehow filter out those molecules that cannot be drugs, those that exhibit a toxic effect. There's an obstacle to filter them by direct way through testing. Determining the toxicity of a compound is very expensive. Cheaper models of in vitro testing on cells or on proteins are often weakly correlated with in vivo experiments on model animals. Preclinical animal testing, and even more so human clinical trials, is the most expensive part of the drug life cycle. So therefore, the pharmaceutical industry is currently focused on using in silico predictive models to reduce the cost of the drug development process. However, attempts to create universal predictive models for toxicity have not been successful. The thing that is only tenths of 1% has data on toxicity. Researchers do not have enough experimental data on toxicity, and therefore often these models do not exceed 1,000 samples. As a result, there is a predisposition to any particular chemotype, but it's not possible to describe the entire chemical space. Therefore, we decided to arrange a hackathon, which included both the collection of data on the toxicity of structures and the construction of predictive models. As input, the algorithm had to accept only the chemical structures in the form of linear smiles representation. After that, the participants were required to vectorize the molecule and use the value vectors for machine learning algorithms. So we discovered interesting patterns from among our participants and obtained good results. The first two places went to teams called MML and PLQSAR, and the MML team demonstrated the effectiveness of the CatBoost model based on fingerprints of ECFP and max keys with the addition of standard ArcticKit descriptors. They showed the high performance for LD50 targets. So BLQSAR teams showed the effectiveness of their CatBoost model on max keys together with ArcticKit descriptors on TOX21 classification task in comparison with known language models. So we see that gradient boosting can effectively solve problems in the presence of a large amount of data and do it no worse than neural networks. So that's why we hypothesized that the most effective solution could be achieved using gradient boosting. Therefore, we took the data sets collected from the hackathon and tested two algorithms: CatBoost on fingerprints and max keys, but without descriptors. And most recently, we received the results on XGBoost when using feature fragments. So we believe the using fragments would give us more fine-tuning solutions. And we can see that we have high performance against benchmarks like TrankGRU, so it's a language model, or CardioTox. We also showed this high performance on regression tasks for LD50 against multitask neural network, which was based on Syntelly website. So these models you can use them on app.syntelly.com. And the last thing I would like to show you that there's a small proof that the more data we have, the better the predictive models we can obtain. And this is very important for chemistry problems. In this graph, you can see the dependence of the RMSE error against the number of samples with logarithmic scale. So here's clear the amount of data is an essential component in obtaining high quality models. So it was our the main goal to show that we should collect the data more and more to get better models. Thank you for your attention.
Thank you, Dmitrii, for keeping the time. And there is some questions from Han Lee. How to know what your method is better than the benchmark
metrics
without some statistical test? It would be better to see if the differences are significant.
Okay, yes. So we have folds. We had 5-fold cross-validation, and these results will be presented on our website, so you can calculate the statistical deviation and evaluate that our model will be better. So we have such small statistical deviations, so that's why we think that it's statistically significant.
Dr. Dmitrii Shulga asked, the proposed models should perform on the vast chemical space based on aspired data sets on which they fit. How do you address unavoidable overfitting, something beyond a straightforward CV?
Okay. So yes, there could be overfitting, and it could be really unavoidable, but actually we don't know what is the molecule would be inspected by this approach, so we can't make our samples, our folds narrow in some of chemotypes. So that's why we can avoid the overfitting by making hyperparameter optimization. So we did it, and also with collecting more data, because it allows us to get more chemical space vision.
Dr. Sivanandam asked, is it free, all this program, software,
so
commercial?
Yeah, okay.
Or commercial?
You can get the token via admin Syntelly.com, so you can write an email, and I guess we can give you a free token for free trial for using our software.
Professor Parokov asked, do you really think that the performance depends only from the number of compounds in the training set?
No, not the only the number of compounds, but also the quality of data preprocessing and the quality of the experiment. Indeed, yes, but there's a trend. So, and there's a main problem in QSAR modeling in chemoinformatics. So there is small data. So there's so many models that have samples lower than 1,000, and they actually can't get good results over the chemical space which training data sets were represented.
Dr. Hanush Senderovich asked, is the method fundamentally different from using the covariance matrix in Cartesian space?
I guess
No,
this question is not for me. This question is not for me.
It was, yes, it was previous. Okay, I'm sorry. I saw the time. So there is no questions, other questions?